Hello Medicose, welcome to the channel.
This video is on microcirculation and the lymphatic system. Today we'll explore how blood moves through capillary beds, how substances cross the capillary walls, and how excess fluid is managed by the lymphatic system. We'll also dive into the Starling forces that regulate fluid exchange and the causes of edema. Let's get started. Capillary beds play a crucial role in microcirculation. Blood enters capillary beds through small vessels called medarterioles, which then branch into capillaries. At the junction of arterioles and capillaries is a precapillary sphincter, a smooth muscle band that regulates blood flow. True capillaries consist of a single layer of endothelial cells without any smooth muscle. Small pores or clefts between the endothelial cells allow water-soluble substances to pass. Blood flow is regulated by contraction and relaxation of arterioles and precapillary sphincters. Lipid-soluble substances like oxygen and carbon dioxide diffuse freely across endothelial cell membranes. Small water-soluble substances such as water, glucose, and amino acids pass through the clefts between endothelial cells. However, protein molecules are too large to move through these clefts, except in specialized capillaries. For example, sinusoids in the liver and intestine allow the passage of proteins, while the blood-brain barrier has exceptionally tight clefts. Larger water-soluble substances can cross by pinocytosis, a process where cells engulf fluid. Fluid exchange between capillaries and tissues is regulated by Starling forces. These forces include capillary hydrostatic pressure, PC, pushes fluid out of the capillaries, promoting filtration. It's higher at the arteriolar end and decreases toward the venous end. It's generated by the blood pressure inside the capillaries and is influenced by both the arterial and venous pressures. At the beginning of the capillary bed, closer to arterioles, the hydrostatic pressure is relatively high, ~30-40 mmHg. This favors filtration. As blood flows toward the venous end of the capillaries, the pressure drops, ~10-20 mmHg, which can favor fluid reabsorption back into the capillaries. Interstitial fluid hydrostatic pressure, pi, is the pressure exerted by the fluid in the interstitial space, the space between cells outside of the blood vessels, against the capillary wall. It plays an important role in regulating the movement of fluid between the capillaries and the surrounding tissue. Pi generally works to oppose the movement of fluid, filtration, out of the capillaries into the interstitial space. If the interstitial fluid hydrostatic pressure increases, it can push fluid back into the capillaries, countering the capillary hydrostatic pressure. Under normal physiological conditions, pi is either close to 0 mmHg or slightly negative, around -1 to 0 mmHg. This low pressure creates an environment where fluid can easily move from the capillaries into the interstitial space. A slightly negative pi can help promote filtration of fluid from the capillaries. If lymphatic flow is impaired, interstitial fluid can build up, increasing pi, which can result in swelling or edema. Capillary oncotic pressure, PC, also known as capillary colloid osmotic pressure, is the force exerted by plasma proteins, primarily albumin, that tends to pull water into the capillaries from the surrounding interstitial space. It is a key factor in maintaining fluid balance between the blood and tissues. Plasma proteins like albumin, globulins, and fibrinogen are large molecules that cannot easily cross the capillary walls. These proteins stay in the blood, creating a higher concentration of solutes inside the capillaries compared to the surrounding interstitial fluid. This difference in solute concentration causes water to move into the capillaries through osmosis to balance the concentration of solutes, thus generating a pressure known as capillary oncotic pressure. Capillary oncotic pressure opposes filtration by drawing fluid back into the capillaries from the surrounding tissue, helping to maintain fluid balance. Interstitial fluid oncotic pressure, PI, favors filtration and depends on the protein concentration in the interstitial fluid. When capillary hydrostatic pressure increases, filtration is promoted, while increases in capillary oncotic pressure promote absorption. Changes in these pressures lead to fluid movement across the capillary wall. Several factors can influence filtration in the capillaries. Dysfunction in the lymphatic system can lead to inadequate drainage, increasing interstitial fluid oncotic pressure, PI, and favoring filtration. The lymphatic system helps return excess filtered fluid and proteins to the bloodstream. One-way flap valves in lymph vessels ensure unidirectional flow, allowing interstitial fluid to enter but not exit. Larger lymphatic vessels also use one-way valves and are aided by skeletal muscle contraction. Edema occurs when the lymphatic system cannot handle the excess interstitial fluid, causing swelling. This can be due to excess filtration or blocked lymphatics. For example, histamine release causes both arteriolar dilation and venous constriction, increasing capillary hydrostatic pressure and leading to localized edema.